Hi, and welcome to the fourth of our five-part lecture series on biomolecules, especially as they relate to the coronavirus. So today we're going to be talking about carbohydrates. This is a topic that we've already touched on in this course. When we think about combustion, it's a pretty likely target. So what are we talking about with carbohydrates? Generally, any carbohydrate is something that's composed of carbon and hydrogen and oxygen, and usually in the proportions shown there. So we have equal parts carbon and oxygen with double the amount of hydrogen. Or if you like, it's a carbon and a water. Again, carbohydrate. In this context, when we talk about it as a biopolymer, we're really looking about sugars. So in all of the polysaccharides or polymerized carbohydrates that we're looking at for biomacromolecule, it's using sugars as the monomer as it stretches out either into a few or many or single chains or branch chains polymers. And how they bond together and which component parts really makes the difference here. So there are shown two sugar molecules that most people are pretty familiar with, so glucose and fructose. One you'll see is a 6-membered ring, one is 5. And how these bond together and play with others really changes what the oligomer or polymer is. So the most common, so our table sugar, most common for us is sucrose. So this is 1 glucose and 1 fructose monomer combined together into a dimer. And you'll also see how they're bonded through these through this alcohol group on the glucose. So it's an ether bond in most of these cases, and how those bond through that oxygen is going to determine whether or not it's something that we can digest and use for cellular respiration or not. So let's look at a few reasons why living things or living organisms would use carbohydrates. So number one is the one that we would most commonly think of. So for energy storage and transportation. Storage, yes, even though we don't produce the carbohydrates, it's what we're taking in and eating and using for our energy. So some of the simple disaccharides, which are made out of monosaccharides, would be things like, we already saw sucrose. Here's maltose. So these are 2 glucose molecules attached through an alpha type bond. And if we switch that up a little bit, so now we've got galactose instead of one of the glucose, and it's bonded through a beta type bond. So you'll see it's effectively the same thing, except now, so this is showing it either as bonded on the same side or opposite sides. You could also think if we switched this galactose and just turned it over, now the location of that, say, look at this CH2OH right here, would be turned upside down, but the bond would be the same. So that's kind of what we're tracking here. And of course, sucrose we've already seen. So this is a fructose and a glucose bonded together. So of course, yeah, so sugar transport in plants. As the plants produce these sugars through photosynthesis, you don't always want it to just be a very available sugar. Because it's going to make you a more attractive target for things that want to eat it. Secondly, they're a lot less stable. So as we bond it into longer chains, it becomes more stable and more easily stored. So that's the goal of most plants, sticking their monosaccharides, so sugar molecules, into starches. When we're storing energy, we don't do it as starch. We do it as what we would call fat, but what is actually glycogen. So here's the difference between a starch, which is just a long single chain of glucose molecules, and where we and animals and fungus and bacteria would instead produce glycogen, which is a pretty highly branched, similar type of polymer. Again, just to store these, store energy, really, that makes it available. We can use an enzyme to break these apart into glucose molecules again, which our body can use for food. So there's energy storage. The other major component, or the other major reason why plants and animals would use polysaccharides, is for structure. So cellulose and chitin are the big ones here. Cellulose is probably the most common biomolecule on the planet in terms of mass. All trees, all plants are using cellulose for their structure. So think of wood when we hear cellulose. There's other proteins attached to that, say lignin, for example. But the polysaccharide cellulose is really giving a lot of that, doing the heavy lifting there. You'll see, now look again at the CH2OH group. So where we had it in starches, they were all on the same side as we had this bond going, again, on the same side through the oxygen bond. Let's compare down to cellulose, where we've got, okay, the CH2OH group is on the upside, we've got the oxygen bond on the same side, but it's alternating where we find that CH2OH group. So this is the opposite type of bonding through that oxygen between the glucose molecules. And this is the only difference between easily digestible starches and impossible to digest cellulose. Chitin, we're going to see there's a small difference here. Well, kind of a major difference now that we've added on an amine and also a ketone group. So this whole thing we can call differently, but we'll look at chitin in a minute. So same kind of arrangement as the cellulose, but we've changed one of the groups attached to that glucose monomer. Okay, so yes, this is what we just said. It's the only difference between starch and cellulose is the type of bonding. Of course, like we say there, if it were easily digestible, it wouldn't be as common as it was. We wouldn't, trees wouldn't be using it for structure because bacteria and animals and, well, other plants and fungus, basically everything, would be eating it and trying to use it to extract the energy. Because that's a lot more difficult to do, it can be used for structure and it can last thousands of years in the ground without being broken down by bacteria. So chitin, this N-acetyl glucosamine, so N-acetyl. So here's where we have an acetyl group. So this would be this ketone group here, attached through the nitrogen on an amine on a glucose. So this would be a N-acetyl glucosamine. Maybe you've heard of glucosamine in terms of like there's supplements you can take to help with joint strength and health, that sort of thing. A lot of the sources for glucosamine in those supplements is from shellfish. Because chitin, so this N-acetyl glucosamine, as a polysaccharide, is used by invertebrates. So beetles, shellfish, squid, octopus, cuttlefish, any of those types of animals, for any kind of hard surface. So we're looking at shells on either crabs or lobsters or beetles. Butterfly wings are made out of the same material. So if you look in very closely, and this is a cool one that I suggest you look up somewhere else, but the blue of these brilliant blue butterfly wings has nothing to do with pigment at all. It's strictly the structure of the chitin that is refracting light in such a way just to make that blue color. So have a look at somewhere else there, because you can really zoom in with these scanning electron microscopes to look at the nanostructure on these wings, and that is what is producing the color, not any kind of other pigment material. Here we also have a squid beak. So I don't know how many people know that squids and octopus have a beak. It's really the only hard surface on them, and that is also made out of chitin. It's a really interesting structure too, because the concentration of water decreases the closer we get towards the tip of the beak, and the water concentration increases as we go away from it. So you can have a really highly refined difference between the hard tip of the beak and surfaces where you'd want to kind of do the pinching, and a softer surface and a softer material as we get further away, where they would want some more flexibility to avoid cracking. Okay, so those are for the structure. How exactly does this play into the coronavirus? Since we're relating everything back into that one nasty organism, how are we going to, build in some carbohydrates here? The viruses, they don't need energy because they're stealing from the cells, and they don't need structure because the only structure there really is the lipid envelope. Well, the other thing that carbohydrates can do is that they can bond with lipids or with proteins to assist with cell, like between the cell interactions, and other tasks when it comes to molecular recognition. So, an easy example to do there are the antigens that we have on our different blood types. So, the difference between, say, O and A, B and AB positive or AB blood types is the arrangement of the glycoproteins, or which sugars are attached onto the proteins on the surface of the blood cell. So these can be recognized by the immune system in the bodies, and why other blood types are rejected. These types of carbohydrates attached to proteins are also called antigens because they will produce or generate antibodies. So this is also a component in our virus. The proteins that we've already talked about, so that spike protein and some of the other ones on the surface of the coronavirus, are quite heavily glycosylated. Or there's between, I think, 69 and 81 different sites on that protein that has a carbohydrate attached to it, to do both to assist with cell recognition and binding, and also to protect it against elements of our generic immune system. But what it does do is produce a good target for producing a vaccine or for other kind of antiviral drugs. If it can target onto those sites where it needs the carbohydrate to protect itself from the immune system, remove that protection, now it's going to be become more subject to our body's defenses. Okay, so we're going to leave it there for carbohydrates, and we'll pick it up next time with our final biomolecule, nucleic acids. Okay, so I'll see you that time, and stay safe.